Hello students, I am teacher Svetlana and in our today's session we will be learning about regulations of gene and the operon concept which is covered in the chapter Molecular Basis of Inheritance. So let's get started. Regulations of gene expression. Students, it is the multi-step process by which a gene is regulated and its product is synthesized. Regulated means the expression of gene is controlled. Controlled means whether it has to express itself or whether it has to suppress or not express itself. Thus, gene expression results in the formation of a polypeptide. Gene expression process is regulated at different levels. In eukaryotes, the regulations can be at different levels. This means that we can control the expression of genes at different levels. Now, these levels are the first one being the transcriptional level, that is formation of the primary transcript. Here I have put an image of the levels of regulation of gene expression. This is your gene on the DNA. Then you have the process of transcription, that is the formation of your mRNA. So when the primary transcript is being formed, at that level also you can control the genes, expressions of genes. Then the next is the processing level, that is when your mRNA is being processed. So where specifically this happens, the regulation of splicing. Just to give you a quick recap of what is splicing, when our mRNA is formed in the eukaryotes, it goes for its further processing, and one of the step is splicing. It is the removal of the non-coding sequences. Now, this is your DNA on which your mRNA is formed. Then we have two regions on the mRNA, that is exons and introns. Exons are the coding region and introns are the non-coding region. So, during the processing, these introns are removed and only the protein coding sequence is kept. So, this is called as splicing, and here also you can regulate the gene expressions. Then the next is transport of the mRNA from the nucleus to the cytoplasm. So this mRNA has, once it's prepared, it is sent to your cytoplasm for the further expression of genes, that is for the further process of protein synthesis. So at that level also you can regulate or control the transport of your mRNA. And then we have the transitional level. Transitional level is the level where the actual protein synthesis takes place. So these are the different levels where you can regulate the expression of genes. Moving further, students, the question is, why are genes expressed? So genes of a cell are expressed to perform different functions. For example, an enzyme beta-galactosidase is synthesized by the bacteria E. coli. It is used for hydrolysis of lactose into galactose and glucose. Now, lactose is a disaccharide and its single units are your galactose and glucose, which are the monomer units. In E. coli bacteria, they do not have lactose in the surrounding. So, if this E. coli does not have this lactose in the surrounding medium as a source of energy, the enzyme beta-galactosidase is not synthesized. So, it's simple. When lactose is there, then the enzymes required to break down this lactose are synthesized, and in the absence of lactose, the enzymes required for them are not synthesized. So, this is an example of regulation of gene. This is how the reaction looks. You have the lactose, which is a disaccharide unit. In presence of water and beta-galactosidase, it is broken down to its monomers, that is glucose and galactose. So here's an image of how lactose metabolism takes place in your E. coli. This is your E. coli bacterial cell here. This is the cell wall. So you have lactose present in its surrounding medium. Now this lactose is its source of energy. It has to enter into the cell so that the bacteria can utilize it. So you have the enzyme galactoside permease, which allows the entry of lactose inside the cell. And once it is inside, you have beta-galactosidase, which hydrolyzes it into its monomer units, that is glucose and galactose. So these are the two enzymes that are here you can see involved in helping the bacteria to utilize and metabolize lactose. So the genes that are coding for the synthesis of these enzymes are controlled by your lactose. So this is an example of regulation of genes. Students, in the process of regulation of gene expression, we can have a positive control and we have a negative control. Positive control. What is this positive control? Certain bacteria like E. coli adapt to their chemical environment by synthesizing certain enzymes depending upon the substrate present. Now, this statement means that E. coli, for its survival, whatever substrates are present in its surrounding, it adapts to it and tries to utilize that substrate for its survival, and then hence synthesizes the enzymes that are required for their digestions depending upon the substrate that is present. Now, such adaptive enzyme is called as inducible enzymes. A set of genes will be switched on when there is necessity to metabolize a new substrate. This phenomenon is called as induction, and a small molecule responsible for this is known as an inducer. This kind of control is called as a positive control. Now, let me explain to you with the help of our same example, that is lactose. Now, when the lactose is present in the surrounding medium, this lactose acts as the small inducer which will allow the synthesis of these enzymes that are required, that is permease, beta-galactosidase. These enzymes that are required for the utilization of lactose are synthesized. Now, how this happens? Have a look at this image here. This mRNA has two regions here. One is the promoter region, one is the operator region. If an activator comes and binds to the operator region, the transcriptions of the genes that will be here, which are required for the synthesis of lactose, for example. So if lactose is the activator, when it comes and binds to the operator region, the genes that are required for the production of the enzymes will be transcribed, and these enzymes, permease, galactosidase, will be produced. And in absence, when lactose is not there, the operator region is free, and that's why you will have no transcription taking place. So such a regulation is called as a positive regulation. Negative control, students, on the other hand, is a repressible regulation of genes, which is seen when the end product of the biosynthetic pathways like the amino acids is provided in the medium. Now, at this time, the internal biosynthesis of amino acids is stopped. So, the amino acid production is stopped, or the metabolite that is being produced is stopped. It is a negative control. So, the metabolite turns off a set of genes involved in the production of the metabolites. So this control is called as feedback repression or a negative control. Now, what this means is when your lactose is being metabolized, you have galactose and glucose being produced. Now, when this glucose, the amount of glucose increases, this glucose goes and now binds to your operator region and acts as a repressor. So what happens is no transcription of those genes will take place. That is, further entry of lactose is then not allowed, because permease will not be synthesized and the process of lactose metabolism will stop. So if the operator is free, the repressor is not there, and activator is there, then the transcription is going to go on. But if a repressor is there, then no transcription will happen. So this is called as, and who acts as a repressor in case of negative control? The end product itself. So here the glucose might act as a repressor. We also have certain other metabolites which act as repressor, and they stop the process of transcription. So this is how the expressions of genes is controlled. Operon concepts, it is a transcriptional control mechanism of gene regulation. Francisco Jacob and Jacques Moudon in 1961 explained that metabolic pathways are regulated as a unit. For example, in E. coli, when lactose sugar is provided to the culture medium, they said that it induces a production of 3 enzymes necessary for its digestion. These enzymes are beta-galactosidase for digestion of lactose to glucose and galactose. Then you have beta-galactoside permease. This permits the entry of lactose into the cell. And you have transacylase, which is required for the transfer of an acyl group from acetyl-CoA to galactoside. So this is how an operon looks. You have the promoter region, you have the operator region, you have the three genes that will form these three different enzymes required for lactose metabolism. We also have the LAC-I, which is going to produce the repressor. So in case of absence of lactose, a repressor is produced which binds to your operator and does not allow these genes to be transcribed. Students, the synthesis of the three enzymes is controlled by a long segment of DNA which is known as an operon. It consists of an operator site O, this is your operator site, and your three structural genes Z, Y, and A. Now the actions of this structural genes is regulated by an operator site. This is the operator site, and this is controlled with the help of a repressor protein. Now, the repressor protein is produced by the action of gene I. It's an inhibitory gene known as the regulatory gene. So, this is your gene I which produces the repressor. The gene expression depends on whether the operator is switched on or off. So, whether the operator is switched on or off. So, there is two possibilities. It is either off or it is in the on condition. Switching on or off of the operator is achieved by the protein called as a repressor. So this is your repressor protein. So what is happening here is when your operon is in the off condition. So this happens in the absence of an inducer. So in the absence of inducer, what happens is your repressor gene produces a repressor. This goes and binds to the operator region. So once the operator is blocked, the RNA polymerase cannot bind here and no transcription of these genes is going to take place. And when your lac operon is in the on condition, this will happen when you have an inducer present. So what is the inducer in case of our lactose metabolism? The lactose is going to act as an inducer. So what this inducer does is when the repressor is produced by your LAC-I gene, this inducer comes in and it binds to your repressor and inactivates it. So the moment your repressor is inactivated, your operator is free, the RNA polymerase can bind here and transcription of these genes will take place. And then further in the translation process, these three enzymes will be synthesized. So this is how the switching on and off is controlled by your repressor proteins. Now students, we need to understand what is a cistron. Each structural gene is generally known as a cistron. So you have this DNA, and every structural gene which will code for a particular protein is called as a cistron. The transcribed long mRNA covering various cistron is known as polycistronic. Have a look at this mRNA. Only a single gene is present which will code for a particular protein. Such a mRNA is called as monocistronic. But if you have multiple genes on the mRNA, like here I have 3 genes, so such an mRNA is called as polycistronic. Now, the further explanation to it is here. For example, in a prokaryotic cell, the DNA, this is the DNA, and these are the coding regions, and these are the non-coding regions. So this will give you 3 different proteins. So such a mRNA is called as polycistronic. But in case of eukaryotes, you will see that you only have the coding region that is come between. Why this happens is because in the eukaryotes, the mRNA is processed and the process of splicing occurs where the non-coding region is removed. So only the coding region is kept which will code for a single protein. So such an mRNA is called as monocistronic. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning.